protection of unvaccinated people when the large majority of the population has been vaccinated. All right, so why am I talking about vaccines in the biotechnology unit? So it is kind of a form of biotechnology. You know, we're using this virus DNA, putting it into chicken eggs. But it turns out the chicken eggs are actually not a great system for making vaccines, especially for some viruses. So how many people remember the swine flu?
Yeah.
It was a while ago, right? It's pretty scary. So it turned out that for whatever reason, the particular strain, the H1N1 strain that caused swine flu, grew really badly in the eggs. So they had a really hard time making enough vaccines to deal with the outbreak. So as a result, the Department of Defense poured a huge amount of money into companies that were researching other methods of producing vaccines. And so one of those methods is with plants. And so this is an approach called transient expression. So rather than genetically modifying plants, say seeds, like we saw before, to make new generations of genetically modified plants, you can actually just insert the agrobacterium into a leaf, and then the leaf will make your protein of interest for a few days after the infection. So it turns out that you can use this to produce vaccines on a pretty large scale. So there's a number of companies now that have these big vaccine production factories where they have tobacco plants that are growing on conveyor belts, and you can come in and spray them with the agrobacterium that has the gene or the virus particle, and then you can produce the vaccine protein in your tobacco plants. And this is a lot faster than using eggs. Everyone? No?
Is that, it's kind of silly, but is that pun like academically recognized?
Yeah, this is what they call it.
That's awesome.
I realize that particularly in Northern California, there's another type of farming. This is not that.
That's hilarious.
So there haven't been any vaccines yet on the market that were produced using this method. But you might have heard about the big Ebola outbreak in West Africa last fall. And so there was a big Ebola outbreak. It was out of control. There were a lot of American aid workers that were over there trying to help out with this outbreak. And these two, a doctor and a nurse from the U.S., became infected with Ebola. They were most likely going to die. And so this company, MAC Pharmaceuticals from San Diego, was making an antibody-based drug that can target Ebola. So what they did was they basically skipped this whole step and just found the antibodies that could bind to the Ebola and alert the body to come in and destroy the Ebola. And they were producing this drug actually in tobacco using that method that I just showed you. And so they applied to the government to let them try this drug out on these folks. So the drug hadn't been tested at all yet. And so normally drugs have to go through a really long trial period. But in cases where they'll die, you can apply for an exception. They did, and both of these folks actually survived. So this is going to be a new methodology of drug production. Yeah.
So that wasn't a vaccine, that was a cure.
Yes.
So they can produce cures as well. And
Yes, you can produce any protein.
Yeah.
Hmm? Yeah. All right, so any questions about vaccines or making drugs and plants? All right, so I think it's time to take a look at how you guys did